It's 11 o'clock, so we're going to go ahead and get started. I'm Matt Dean with A+ College Ready. I want to thank all of you for being here today. We are recording this session. We'll post the video to the A+ College Ready Science YouTube channel. All right, can you hear me now? Can everybody hear me now? All right, great. Somehow I muted myself. So I don't know how much of that you guys missed. So let me go back. I will be sending a copy of the PowerPoint presentation and the free response question out to teachers. And our next webinar is Friday at 2 o'clock. That one's going to be on gene regulation. We'll be talking about things like operons and different ways that both prokaryotes and eukaryotes regulate their genes. That's a confusing topic for a lot of students, so that might be one you want to join us for. But today we're going to be talking mostly about Mendelian kinds of genetics. I've tried to pick out a few topics that students tend to struggle with and things that tend to get asked a lot on AP exams. I've got 2 FRQs that we want to make sure we go over here toward the end. So first of all, I'm going to pull up a different PowerPoint. Give me just a second. So we don't know exactly what the questions are going to look like on this year's exam. Hopefully by the end of the week, by Friday, we'll know. Here are some questions that we've seen in the past. Just quick kinds of things that students ought to be able to do really easily. So I just want to make sure you know how to do them. We won't spend a lot of time on these problems, but I want to make sure you can quickly do these calculations. So first of all, how many different possible gametes can an individual with the following genotype produce? Now remember that each gamete is going to have only one copy of each kind of gene. Your normal cells, your somatic cells, are diploid. Those cells have 2 copies of each chromosome and therefore 2 copies of each gene. But meiosis is going to half that. Meiosis is going to create haploid gametes, meaning those gametes have only 1 copy of each chromosome, or in this case, each gene. The way those genes end up getting distributed is determined by things like independent assortment in meiosis. But essentially what you need to know is that for the A genes in this situation, there are 2 different options, either a dominant big A, recessive little a. Same for the B genes, same for the D, same for the E. So when you're determining possible gametes, remember that each gamete can get a random combination of each A, each B, each D, each E. We're assuming that none of these genes are linked. We're assuming they're all on different chromosomes. So the easiest way to do this calculation is to look at how many options there are at each gene. At the A gene, there's a dominant and a recessive option, so 2 options. At the B gene, also two options, dominant and recessive. Same at the D, same at the E. So to calculate the number of possible gamete combinations, all we're going to do is do the product rule. We're going to multiply 2 times 2 times 2 times 2 to get 16 different combinations. So this individual can make 16 distinct kinds of gametes, each one having 1A, 1B, 1D, and 1E gene. Just an example, one gamete might have all dominants, for example. Another might have all recessives. Another might have some weird mix, like that, for example. But what I'm saying with my calculation is there's 16 different possible combinations that look something like this. So here's the same question with a different setup on B. So again, let's look at the different options. For the A gene, there's two options, dominant or recessive. For the B genes, there's only one option. Every gamete is going to get a recessive allele. For the D genes, two options, either a dominant or a recessive. And then for the E genes, again, only one option. Every gamete is going to get a recessive allele. So again, we're going to use the product rule, and we're going to multiply 2 times 1 times 2 times 1. This individual can make 4 distinct kinds of gametes. And we can quickly write those out. Let me make some space up at the top. So this individual could make a dominant A, a recessive B, a dominant D, and a recessive E. That's one possible gamete. Could also make dominant A, recessive B, recessive D, and recessive E. Additionally, could make recessive A, recessive B, dominant D, recessive E. And then the final one would be this. So those are the 4 possible gametes, haploid gametes created by meiosis through processes like independent assortment and segregation. Now, another kind of problem we've seen on AP exams in the past is something like C. So what is the probability that the cross between the following genotypes, so the following parents, these are our parents, what's the probability that those two parents will create an offspring with this genotype? So you could do a huge tri-hybrid cross Punnett square. Don't do that. Never do that. The easiest way to do this is to think of this like 4 different Punnett squares. And in most cases, you can probably visualize these without drawing the Punnett squares. I wouldn't recommend drawing them. I'm going to draw them to show you what I'm thinking, but in most cases, you can visualize them and go from there. So let's first of all think about the A genes. So I want to know, what is the probability that this and this create an offspring with this genotype? So I could quickly sketch out a Punnett square between these two parents, and it would look like this. And what I can see, the probability of these two parents creating an offspring that's big A, little a is 2 out of 4, or 1/2. I'm going to write that down right here. Now I want to go to the B genes. So I want to know, what are the chances that this and this create this? So again, I'm going to draw out the Punnett square. Hopefully you get to the point where you can just visualize this without drawing it. Here's what the square would look like, though. And we can see again, there's a 2 out of 4 chance that this parent and this parent create that child. 2 out of 4, I'm going to reduce that to 1 out of 2. I'm going to write it down right there. Now I want to know, what are the chances that this and this create this? So again, quick sketch of the Punnett square. And what I can see now is there's a 1 out of 4 chance that the 2 D parents create this genotype offspring. So I'm going to write that down right there. And then finally, let's do the E's. So again, I'm looking at this set of E's and this set of E's and the chances that they produce that. So what I can see is there's a 2 out of 4 chance, a 1 out of 2 chance that we get this genotype. So now I want to think about the whole offspring. What are the chances that this parent and this parent create this offspring? And the way that I'm going to do that is I'm going to use the product rule. I'm going to take the individual probabilities from each gene set and just multiply them across. So I'm going to go 1/2 times 1/2 times 1/4 times 1/2. And that's going to give me, let's see, 4, 16, 1 out of 32 chance that parent 1 and parent 2 create a child with big A, little a, big B, little d, big E, little e. A 1 out of 32 probability. All right, so you might see those kinds of questions. Again, we don't know what the exam's going to look like just yet. By Friday, we should know a whole lot more, and we'll be able to tell you more from there. I want to go ahead and proceed and talk a little bit about pedigrees and modes of inheritance. That's going to be one of our big topics for today. And then we're going to look at FRQs at the end. So you guys, I hope, have all seen pedigree charts, and you know they're diagrams that kind of show us a lineage or genealogy, and they show us how a trait has been inherited across generations. I'm going to talk a lot about the modes of inheritance today. These are the ways in which a trait or a gene is passed from generation to generation. I'm going to talk about 5 main types. I'm going to talk about autosomal dominant. First of all, you need to know that an autosome is a non-sex chromosome. Us humans all, regardless of sex, have 44 of those in every diploid somatic cell, 44 autosomes. So if a trait is autosomal, males and females should be affected pretty much the same way. So we've got dominant versions of those. We also have autosomal recessive traits we'll talk about. We have X-linked dominant and X-linked recessive traits. So we know that males, human males, have an X and a Y chromosome. Those are our sex chromosomes. Females have 2 X chromosomes. So X-linked traits, sometimes called sex-linked traits, are traits for which the genes are located on the X chromosome. And because they're on one of the sex chromosomes, these traits can affect males and females differently. We'll also talk about Y-linked traits. Those are traits whose genes are located on the Y chromosome. There aren't many of those because the Y chromosome is very small, doesn't have a lot of genes. But again, that trait, if it's Y-linked, will affect only males. The final one we'll talk about today is mitochondrial inheritance. Remember that the mitochondria have a very small bacteria-like chromosome. This chromosome seems to be almost always inherited from the mother. And because of that, it can affect children in a certain pattern. Later on today, we'll also mention gene linkage, not so much in terms of modes of inheritance, but we're going to see that genes are linked if they're found on the same chromosome. If they're found on the same chromosome, they're oftentimes inherited together, and that can cause some weird patterns in our Punnett squares. So let's talk briefly about pedigrees. Remember that the symbol for a male is just a square. A female is a circle. When individuals mate, they're connected through the center by a horizontal line. The children of those parents are going to come down from that horizontal line with a vertical, like so. Horizontal. And then anything connected to this horizontal directly is a child of these two parents. You might see twins, dizygotic twins, so these are fraternal twins, non-identical, are drawn with this kind of symbol. And then identical twins or monozygotic twins are drawn with that kind of symbol. Some other things that we know: if an individual is affected with a trait, typically the shape is going to be completely colored in. We also can see carriers carriers are usually shown as either half shaded shapes, or sometimes they're shown with just a dot in the middle, like so. So those are heterozygote carriers. Something that you need to make sure you know is that in some pedigrees, the carriers are going to be marked. In others, they're not marked at all. So they may be a carrier. These may not be indicated. If you put a slash through an individual, that typically means they're dead. They've just been deceased. This essentially means there's incest going on. All right, so let's talk a little bit about the modes of inheritance. So autosomal dominant traits are traits that are, the genes are located on an X chromosome. An individual only needs one copy of that dominant gene, dominant allele, to have the trait. With these kinds of traits, males and females are equally likely to have the trait because the gene is on an X chromosome. These traits don't skip generations because they're dominant. You can't be a carrier for these traits. Whenever an individual has the allele, they have the trait. There can be male-to-male transmission, and what we mean by that is that fathers can pass this trait on to their sons. Fathers can also pass it on to their daughters. Mothers can pass it on to their sons. Mothers can pass it to their daughters. The reason we mention this is because in X-linked traits, fathers can't pass that on to their sons, at least not normally. So there's several human conditions that we talk about in a regular AP Bio class that are autosomal dominant types of traits. Huntington's disease, which is a neurodegenerative disease, is carried on this way. Achondroplasia, probably the most common type of dwarfism, is inherited this way. Polycystic kidney disease is a really common kidney disorder. And then familial hypercholesterolemia, a form of genetic high cholesterol, which can lead to heart disease, that's also an autosomal dominant trait. Here's an example of what a pedigree might look like for an autosomal dominant trait. So here we're looking in the first generation. The male, in this case, is affected with the trait. The female's not. Now, we don't know if this male is homozygous dominant or heterozygous. We don't know. Could be either one. But what we see, the children of this individual, I'm going to connect that with red here. So anybody that's connected to that red line is a child of persons 1 and 2. And notice that three out of the 5 children are affected with the disorder. Notice that individual number 6 and individual number 3, they don't get the trait. That's possible. Also notice that males and females are both inheriting this condition from the father. Individual 7 is the mate of individual 8. Individual 4, the mate of individual 3. And individual 2 is the mate of individual 1. So when you're looking at autosomal dominant traits, what you tend to see, number 1, is the traits don't skip generations. There are people in the first generation that have it. There are people in the second that have it. There are people in the third that have it. There are no carriers. So that's the first thing you want to look at. Does the trait skip generations? In this case, no. Are there carriers? Again, in this case, the answer to that is no. And then the other thing you want to look at are males and females affected pretty much equally. So just count the number of affected males and females. In this case, there's 3 affected males, 4 affected females. The numbers are close to equal. And if that is true, that tells you that the trait is probably autosomal. So this trait, autosomal dominant. We'll talk more about the pedigrees here in just a minute. Let's move on and talk about autosomal recessive traits. So in this case, the only way to inherit a recessive trait, to actually show the trait, is to be homozygous recessive for the trait. So individuals have to have 2 recessive alleles to possess the trait. Because it's autosomal, Males and females are equally likely to have the trait. These traits often skip generations, and they can do that because individuals that are heterozygous, individuals that are carriers, they're not affected. Only homozygous individuals, homozygous recessive individuals, have the trait. This trait can actually appear in siblings without their parents actually having the trait, because the parents could both be carriers. And if that's true, there's a 1 in 4 chance that they'll create children that are homozygous recessive and possess the trait. If a parent has the trait, those offspring who don't get it are at least carriers. For example, let's say we had these two parents. Every offspring of these two parents is going to be big A, little a, which means all of the children would be carriers for the trait. There are several human genetic conditions that are transmitted through this autosomal recessive mode. Tay-Sachs disease, which is common in certain subgroups of the Jewish population. PKU, which is a very common, has to do with digestion and breakdown of certain amino acids, phenylalanine, and then cystic fibrosis, fairly common in the Caucasian population. It affects the respiratory system, but it also affects all parts of the body. It's a systemic kind of infection. Let's look at an autosomal recessive pedigree chart. So the first thing we want to look at, number one, does the trait skip generations? And we can see in this case, there are no affected individuals in the first generation or the second, but there are people who are affected in the third generation. So it does skip generations. If a trait skips generations, it's recessive. Also note there are carriers that are marked. These individuals are carriers. If there are carriers, the trait is recessive. And then finally, we want to look at are males and females affected equally. And we can see we have one affected female, one affected male, and we have both males and females that are carriers. If that's the trait, if that's the outcome that you get, if males and females are affected pretty much evenly, more than likely the trait's autosomal. So this is an autosomal recessive pedigree chart, more than likely. Let's move on and talk about sex-linked traits. And again, these are traits that are determined by genes located on the sex chromosomes, usually either the X or the Y. We're going to talk about X-linked and Y-linked. X-linked are the ones we talk about most of the time, because the X is a normal sized chromosome with lots of important genes on it, whereas the Y is really small and only possesses just a few genes. So let's start off by talking about X-linked dominant traits. So lots of times we'll use a symbol like this. So we'll draw it when we're doing like Punnett squares for an X-linked trait. We want to show both the X and Y chromosomes. Maybe like so. And let's say that this individual has the trait, but it only has one affected allele. So this X chromosome has the dominant X-linked allele we're looking for. This one doesn't. And let's say the father over here, let's say he doesn't have it at all. So his X does not have the dominant allele. His Y doesn't get a superscript at all because this is an X-linked trait. It has nothing to do with the Y. In terms of X-linked dominant traits, though, females are actually a little more likely to inherit the trait because they can inherit it from either parent. They get an X chromosome from mom and from dad. The trait's present whenever the allele is present. So if you have this allele, you have the trait. There can be no male-to-male transmission here because what makes a male is that he got a Y chromosome from his father and not an X. So men can't pass this to their sons. Now, there is a couple of really uncommon exceptions. For example, there are individuals who can end up being XXY, and they could end up getting an X chromosome from their father and a Y at the same time. But I think that's beyond the scope of what you're probably going to be asked about on the AP exam. A female who has the trait can pass it on to a son or a daughter. Doesn't have to. We saw this individual The daughter could inherit this X. The son could inherit this X and not be affected. Or on the other hand, either child could inherit this X and be affected. All of the daughters of a male with the trait will inherit the trait. Because remember, for a guy to have a dominant trait, he has to look like so. And for him to make a daughter, the daughter has to get this chromosome, which is affected, has that dominant allele on the X chromosome. So there's a few human genetic conditions that are transmitted through the X-linked dominant route. Hypertrichosis, which is sometimes called the Wolfman syndrome because of a really hairy body appearance. Porphyria, which is a blood disorder that was common in the royal families of England. And then Rett syndrome. We may talk about that one later. But here's an example of what an X-linked dominant pedigree chart might look like. Again, first thing you want to look at, does the trait skip generations? And we can see affected individuals in each generation. So because it doesn't skip generations, that tells us it's probably a dominant trait. Also, we notice there's no carriers marked. There don't seem to be any carriers. That also leads us to believe that this trait probably dominant. The next thing you want to look at are males and females affected in equal numbers. And if you look, there are 1, 2, 3, 4, 5, 6, 7 affected females, but only 2 affected males. So if females are affected more than males, or if males are affected more than females, more than likely the trait's going to be X-linked. Now, another thing we can look at is who can a parent pass it to? So we can see here, this mother is passing it to both daughters and sons. But then if we look at this affected male, he's only passing it to daughters. His sons here and here are not affected. So that's another clue that the trait is probably X-linked. Fathers can pass it to daughters. They can't pass it to sons. Mothers can pass it to both sons and daughters. Let's move on and look at X-linked recessive traits. So for an individual to get an X-linked recessive trait, for a female, they have to inherit that allele from both parents. For a male to get it, he only inherits it from his mother, because the only X he has came from his mother. He only needs one copy of that allele to have the trait, because that's the only X chromosome that he has. For that reason, these traits are more common in males than females. A male only has to inherit that recessive allele from one parent, whereas a female has to inherit that same recessive allele from both parents. Since these traits are recessive, they often skip generations. All daughters of a male who has the trait are either affected or are carriers for the trait. Because remember, the guy has to have the allele. His daughters are always going to get that allele. So they're always going to have one copy, at least, of the recessive allele. The son of a female carrier has a 50% chance of having the trait. So a female carrier would look like this. The son's either going to inherit this and not have the trait, or he's going to inherit this and have the trait. Mothers of males who have the trait are either heterozygous carriers or homozygous and express the trait. So again, a female, the mother of a son who has the trait. Here's a son who has the trait. The mother had to be either a carrier, like so, or she herself had to be affected. That's the only way the son gets that genotype. As with X-linked dominant traits, there's no male-to-male transmission. Males can't pass these traits on to their sons, except for the weird, the rare exceptions to the rules. There's several human genetic conditions that are transmitted via the X-linked recessive mode. These include hemophilia, which is a blood clotting disorder, the most common type of color blindness, red-green color blindness, the most common type of muscular dystrophy, Duchenne muscular dystrophy, which affects muscles and they degenerate, and then also Lesch-Nyhan syndrome. All of these traits, though, are way, way more common in males than they are in females. Here's what a pedigree might look like for an X-linked recessive trait. So again, first thing we're going to look at, does the trait skip generations? And we can see in generation 2, even though there are some carriers, there are no affected individuals. If a trait skips generations, that should tell you it's probably recessive. The other thing we want to look at, are there carriers? And there are. So if they're carriers, the trait has to be recessive. The next thing we want to look at are males and females affected equally. And what we can see is there's lots of affected males, only one affected female. There are lots of females that are carriers, but almost no affected females. So if males and females are affected differently, it's sex-linked. In this case, since a female can get it, we know that it has to be an X-linked recessive condition. Another thing you want to notice here is that the only carriers are females. If you have a trait where the only carriers are females, it's going to be an X-linked recessive trait. Moving on, we'll go through this one really quickly. Here's a Y-linked trait. Only males are affected because females don't have Y chromosomes. All sons of an affected male will also be affected. Let's say that's an affected male. The reason his sons are his sons is because they inherited this Y chromosome. Every son of this male will have this condition. Females don't possess or pass on the traits. There's several types of disorders transmitted this way. There's a retinitis pigmentosum, which is a progressive type of a blindness. There's also this condition that causes really hairy ears, and it is also transmitted via the Y chromosome. Here's what a Y-linked pedigree chart might look like. We don't talk about these in terms of dominant or recessive because there's only one Y chromosome. So essentially everything's dominant. Notice that for this affected father, every son that he fathered has the trait. Person 7 here is not his son. That's his son-in-law, not affected. Again, notice person 4, both sons have it. Person 8, the one son has it. This can't be passed on to daughters. So if you see a pedigree where men are passing it to only men and to every male child, it's going to be a Y-linked trait. The final one of these we'll talk about is mitochondrial inheritance. Remember that mitochondria has non-nuclear DNA, a small chromosome. It looks like a bacterial chromosome. These seem to be passed on always from mother to child, not from father to child, and from mother to every child. So there's really no dominant or recessive mitochondrial traits because the mitochondria only has one chromosome in it. So if you get the trait, if you get a copy of the allele, you have the trait. Individuals who possess a single copy of the allele express the trait. So what we're going to see in mitochondrial types of pedigree charts is that all of the children of an affected female have the trait. Males can have the trait, but they don't pass it on. There are lots of disorders that can be passed on for this route. Many of them have to do with metabolism and cell respiration. So here's an example of a mitochondrial pedigree. Notice this female has it. Both of her children get it. This affected male passes it on to no one. This affected female passes it on to every one of her children. This male passes it on to no one. This female passes it on to every child. So those are mitochondrial pedigree charts. Those are usually pretty easy to pick out. All right, so here are a few pedigree charts that we want to go through and determine the mode of inheritance on. So I want you guys to take just a minute, and I want you to take A and B, and I want you to try to determine the mode of inheritance. I'll give you like a minute and a half, 2 minutes to try to determine the mode of inheritance on A and B. All right, I want to move through these fairly quickly. So again, the first question you want to ask is, first of all, does the trait skip generations? And we see in A, there are affected individuals in every generation. So that hints us in this might be a dominant trait. The next thing, are there carriers that are marked, or do there seem to be carriers that aren't marked? And again, that doesn't seem to be the case in this pedigree. So again, that leads us to believe this is probably dominant. The trait isn't skipping generations. There don't seem to be any carriers. And the next thing you want to look at are males and females affected equally. So just count the number of affected individuals of each gender. So 1, 2, 3, 4, 5 affected males. And there are also 5 affected females. If the genders seem to be affected equally, more than likely the trait is autosomal. So my best guess on this would be autosomal dominant. And one thing that would also prove that is notice that this father right here passes the trait on to a daughter, but he also passes it on to a son. If males can pass it to both parents or both children, both genders, it's more than likely an autosomal trait. Let's move on down to B. So again, first thing we're going to check on, does the trait skip generations? Do there seem to be any carriers? And our answer to both of those questions is no. So again, that hints us in this is probably dominant. Next thing we want to look at is, are males and females affected equally? And what we can see in this pedigree in B is that there are a lot more affected females than there are males. If that's the case, if one gender is affected more than the other, it's probably going to be a sex-linked thing. And since females can be affected, it can't be Y-linked. So this is going to be an X-linked dominant trait. Other things that lead us to believe that's true, notice that males are only passing it on to females, and they're passing it on to every daughter. Affected males are passing it to every daughter. Whereas females, like this one, for example, can pass it to sons. Females can also pass it to daughters. If that's true, it's going to be an X-linked trait, and in this case, X-linked and dominant. Let's go ahead and look through C together. Again, first question, does it skip generations? Are there carriers? This one skips the first generation, and there are carriers marked with dots. So if that's true, either one of those is true, that's going to tell you the trait's probably recessive. Next question, are males and females affected equally? And we can see that only males in this pedigree are affected. Now, you might think Y-linked, but this can't be Y-linked because females are carrying it. So this trait has to be an X-linked recessive kind of thing. X-linked recessive. Let's go on down and look at D. So again, we see that generation 2 is completely skipped. There are carriers. This has to be recessive. We also see that males and females seem to be affected equally. That would lead us to believe this is autosomal. And another bit of evidence that would prove to us this is autosomal is that males can be carriers. If males can be carriers, it can't be sex-linked, because males only have one of each sex chromosome. So the mode of inheritance on D, autosomal recessive. Got a few more we'll run through real fast here. On E, check to see if it skips generations. It does not. Check to see if there are any marked carriers or if there seem to be any unmarked ones. Doesn't look like it. So that leads us to believe this is recessive. Next, look to see if males and females are affected equally. And they are not. There seem to be many more females than males. That's the case. That's going to hint us in this is X-linked, and in this case, X-linked recessive. And again, a bit of proof. Females are passing this on to both males and females. Males can only pass this on to daughters and not to sons. More proof that this is X-linked recessive. Going on to F, this trait skips generations. There are carriers that are marked. That tells us it's recessive. Males and females are both affected equally. That tells us it's probably autosomal. Males can carry the trait, so that tells us it can't be X-linked. So autosomal recessive looks good here. On the next one, doesn't skip generations. There are no carriers. That tells us probably dominant. Let's look at the male to female ratio. So there's 3 females affected there, plus 1 there. 4 total affected males as well. That tells us probably autosomal. Another thing that shows us autosomal is probably right is that males can pass it on to sons. Females can pass it on to sons. Females can pass it on to daughters as well. We don't see it here, but we would assume that males can also pass this on to daughters. All right, the last one. Again, we see that this trait skips generation 2 completely. There are carriers that are marked, so that tells us it's recessive. We see that males are the only affected individuals, but that females can be carriers. So that eliminates Y-linked. That tells us that this trait has to be an X-linked recessive condition. All right, those are our pedigree charts. Let me take a minute and answer questions if there are any. Anyone have any pedigree chart questions before we move on?
Yes, I was wondering, how is E recessive?
One other quick topic I'll mention before we get to an FRQ is phenotypic plasticity. Phenotypic plasticity occurs when individuals with the same genotype have different phenotypes because they're in different environments. The environment can affect gene expression. Apparently, I had a question and I didn't hear it. I'll come back to that in just a minute. Let's talk about the phenotypic plasticity, though, for a second. A good example of this is the Arctic fox. They tend to be red and brown in the summer months, but they turn pure white in the winter. Turns out that the way this works is the genes that produce the red-brown pigment are blocked by cold temperatures. So they're inhibited by cold temperatures, and that causes the hair to grow with no color. Another example of this is the color in hydrangeas, a type of flower. In certain pH soils, those flowers are blue, where in other pH soils, they're pink. Another example, the diets, especially protein-rich diets in adolescence, tend to cause taller heights in the US and Europe than in more famine-stricken places. So don't want to spend a lot of time on that, but we'll come back to it again in a minute. All right, I'm going to get out of here for a second. Let's see if I can get the question. So I couldn't hear the question, so if you guys will try again, and let's see if I can't get your question.
I was wondering, the example E, it was recessive, but it didn't skip generation. So I was curious to see why it was recessive.
You said E?
Yes, sir.
Let me go back there for just a second. Yeah, so I said recessive on that one?
Yes.
I just misspoke because that one is not skipping generations. There are no carriers, so that means it's going to be dominant.
All right, thank you.
Yeah, so it's X-linked to dominant. I just misspoke on that one. Thanks for good catch. Any other questions on that one? All right, what I want to look at for the rest of our time today are FRQs, and that's really going to be the focus of most of these sessions. I went a little longer today than I wanted to on the content. So let's take a minute and look at this FRQ. I'm going to try to blow it up a little bit so you can see it. So if you guys will take a minute and just read through this, I'll scroll down so you can see the rest of it in a minute. This document has also been sent out to your teachers, so you can get a copy from them just by emailing them. But take a minute and read this, and I'll slowly scroll through it as you're reading. All right, so what we want to focus on first here, just part A. And some of you have already been in one of these webinars with me, so you know sort of my approach for FRQs. When you're answering an FRQ, I want you to write down the question number on your paper and part A. And I only want you to answer part A. I want you to make sure that you answer the prompt. We like to use the acronym ATP. Answer the prompt. Don't say anything else. Just answer the question. There's no need to talk in circles and throw fluff in there. Get to the point, answer the question, pay specific, pay attention to the verbs. They're always going to be in bold. That's where your points come from. Answer the prompt. Each one of these pieces, probably 2, 3, at the most 4 sentences is all it's going to take. You don't need to do an introduction, a body, and a conclusion. You just need to do a body. Answer the question, move on. ATP, address the prompt. So again, first question, what conclusions can be drawn from cross 1 and 2 and explain how the data support your conclusions? They're going to give you data on all these questions. They're expecting you to talk about that data when you answer the questions. So again, explain using the data. That's important. So let's look at cross 1 and 2. Let me scroll back up a bit. And I do these individually. So we're looking at fruit flies here, or some kind of fly. One thing I want to make sure that you know, if they talk about true breeding means if you took 2 of the individuals and you bred them together, you would get identical individuals for that trait. What true breeding is really another word for is homozygous. So if they tell you an individual is true breeding, that means for that trait, it's either homozygous dominant or homozygous recessive, one or the other. Just get in your mind, true breeding means homozygous. So let's look at cross 1. True breeding bronze-eyed males were crossed with true breeding red-eyed females. All the F1 offspring, that means their babies, the babies of these individuals, had bronze eyes. All right, so let's think about what that means. So we have these males, bronze-eyed, crossed with females with red eyes. All of their babies have bronze eyes, regardless of sex. So what I'm hoping that tells you is, number one, since they had to have gotten a red allele from mom and a bronze allele from dad, I'm hoping that tells you that bronze is dominant. And also what I want you to think about is that both the males and females got bronze. It didn't seem to matter what sex they were. Males and females seem to be affected the same way. So that's a good idea that this is probably also an autosomal trait. We'll come back to that in a second, though. Let's look at the F2 phenotypes. So in the F2s are the babies of the F1s. So the babies of these bronzes. Let's think about some symbols for a second. Let's assume for a minute that this trait is autosomal. And let's say that the original father, let's use big B for bronze, and let's use little b for red. You could use any symbol you wanted to use. If those are the original parents, what we might call the P generation, all their children are going to have one big B from dad and one little b from mom. All the children are going to be heterozygotes. So these are the F1s. If we crossed 2 F1s, and that, by the way, that's how you get the F2 generation, is you cross 2 of the F1s. So if we crossed 2 of those, and we did a Punnett square for that cross, what I'm hoping that you would see for that Punnett square is that we would expect these 3 to all be bronze and this one to be red-eyed. So we're expecting a ratio of 3 bronze to 1 red, assuming it's autosomal. So let's look and see if that's what turns out in the F2. First of all, notice males and females have about equal numbers of bronze eyes, and males and females have about equal numbers of red eyes. Again, good clue that this is probably autosomal. Let's combine these numbers roughly. So let's just round this off to 3,700 and 3,800. That'd put us at about 7,500, right? Exactly, it's 7,520. And then let's add these two together, 1,260 plus 1,320. So that's 1,580. And notice that ratio, although not exact, and that's 2,580 instead of 1,580, not exact, but pretty close, is a 3-to-1 ratio. Again, more proof that this is probably an autosomal recessive trait. So if I were answering this question about part 1, my conclusion would be that bronze eyes are autosomal dominant. Red eyes are autosomal recessive. And the way that I'm going to back that up with my data is I'm first of all going to say all of the F1 offspring, regardless of gender, were bronze. That tells me the trait of bronze is probably dominant. Number 2, I'm going to say that in all generations, males and females were affected in essentially equal numbers. Equal numbers of bronze-eyed females as there were males. Equal numbers of red-eyed males and red-eyed females. Almost equal. Again, that tells us probably autosomal, and probably autosomal dominant for bronze and autosomal recessive for red. I'm going to skip over to the scoring guidelines for this real quick, show you what that looks like, and then we'll come back and look at cross 2. So here's what the actual graders use to grade cross 1, this box. And what they're wanting you to say is, number 1, bronze is dominant, red is recessive. Get a point for that. You also might earn a point for saying that it's not a sex-linked trait. And then you get a point for why. All the F1s expressed the dominant bronze trait. The F2 show essentially a 3-to-1 ratio. There's equal distribution of F2 phenotypes for both genders. If you said one thing out of this box and you backed it up with one thing out of this box, you got 2 points for cross 1. Let's go back and look at cross 2. Same kind of reasoning. So again, true breeding normal winged males were crossed with true breeding stunted winged females. So these females have little bitty tiny wings that don't work. The males have normal wings. And again, true breeding means that the original parents are homozygous for those traits. All the F1s had stunted wings. So we're taking normal wings, crossing with stunted wings. These are both homozygous. So we know in the these are the parental generation. We know the F1 get one normal wing gene and one stunted wing gene. They all turn out to be stunted. So I'm hoping that leads you to believe that stunted is probably dominant. Then let's look at the F2s. Notice the F2s. Male and female normal wings, similar numbers. Stunted wings, male and female, similar numbers. So similar ratios. Again, that leads us to believe that stunted is probably dominant. And since the male to female ratio is pretty much the same, that leads us to believe that it's probably also autosomal. So I would also say that stunted wings are probably an autosomal dominant trait. And I'm just going to abbreviate autosomal and dominant, which means that normal wings are autosomal and recessive. And the data that tells us that is, number 1, all the F1s, regardless of gender, have stunted wings. That tells us it's probably dominant. And then in both generations, the F1 and the F2, Males and females have essentially equal distributions, equal ratios. That tells us the trait is probably autosomal. We could draw a Punnett square for this one as well, and I'll do that real quick. I'm going to use an S, a capital S for stunted. So we had that stunted mother crossed with a normal winged father. We'll use little s's to mean normal, and I'll put a line under those so you know they're little. That's the parental generation. In the F1, all of the offspring had to be heterozygous. This is the F1. To make an F2, we had to cross two of these F1s. So big S, little s, crossed with big S, little s. Our Punnett square is going to look something like this. So what we're seeing is that these three should all have stunted wings, the dominant trait. This one should have normal wings, the recessive trait. So we're expecting a 3-to-1 ratio in the F2. And if we look at those numbers, here we have, that's, sorry, I'm on the wrong one. So here for stunted, we have 3,600 plus 3,820. So that would be, what, 7,420? And then here we have 1,160 and 1,320. So let's see, that's going to be 2,480. Not a perfect 3-to-1 ratio, but really, really, really close. So let's go down to again, the scoring rubric for this. And by the way, we're going to go through the rest of this question and then we'll call it a day. And we'll do the other FRQ during the next session on Friday. So I need about 5 more minutes to finish this up, guys. So here's cross 2. Here are the scoring guidelines for cross 2. What they want you to say for 1 point is that stunted is a dominant, normal is recessive. And the way you know that is that all F1 heterozygotes express the dominant stunted trait. So you got a point for saying this and a point for backing it up with the data. They also want you to say that stunted is autosomal. It's not sex-linked. And you know that, backing it up by the data, by saying the F2s have essentially a 3-to-1 ratio of stunted to normal. Or you could have said that there's an equal distribution of the phenotypes for both genders. Either one of these would have given you a point there. So again, 4 possible points on part A. Fairly easy to get if you make conclusions and then back that up with the data. That's important. Refer to the data. If they give you a graph, if they give you a chart, if they just give you some text-based information, when you're answering your questions, you need to refer back to that data. All right, so the last part of this is what conclusions can be drawn from cross 3. Let's scroll down and look at cross 3. So we've got true breeding, bronze-eyed, stunted winged males crossed with true breeding, red-eyed, normal winged females. So let's think about what that means, first of all. We know that bronze is a dominant trait. We established that in cross 1. This first parent is true breeding bronze. We also established that stunted wings were dominant. This male is true breeding for both of those. So that would be our first parent. Our second parent, so we're crossed with true breeding red-eyed. So we assume that red eyes are recessive and normal wings recessive. All F1 offspring had bronze eyes and stunted wings. Well, that would make sense because every offspring should get a big B from dad, a little b from mom, a big S from dad, and I use the N's here. These should be S's. And little S's, a little S from mom. So that should be the phenotype for every offspring in the F1. And that would be bronze and stunted wings. So that makes sense. That goes with what we said in crosses 1 and 2. The F1 flies were crossed with a true breeding, red-eyed, normal-winged flies. And the results are shown below. Well, let's think about that for a second. So we're saying we took one of these F1 flies, big B, big S, little s, and we crossed that with a true breeding red-eyed female or true breeding red-eyed individual. The only way to have red eyes is to be homozygous recessive. It's a recessive trait. The only way to have normal wings is to be homozygous recessive. So to make the F2s, or to make this generation of flies, we crossed these individuals. So we need to do a Punnett square real quick for this. And I'll do that over here. What we would expect in the Punnett square, this individual has 2 different B combinations, 2 different S combinations. So that's telling me they ought to be able to make 4 distinct kinds of gametes. Those gametes would be a big B and a big S, a big B and a little s, a recessive B, a dominant S, and a recessive B and a recessive S. Whereas this individual only has 1 B option and 1 S option, which is telling me this individual, really all their gametes are going to be the same. Every gamete is going to have a little b and a little s. So if we draw out our Punnett square, some individuals should be this genotype, and that's going to be bronze and stunted, right? Some are going to be this, and that's going to be bronze and normal. Some should be red-eyed and stunted, and then some should be red-eyed and normal. So we're expecting 1/4 of each possible phenotype. 1/4 bronze stunted, 1/4 bronze normal, 1/4 red stunted, 1/4 red normal. That's assuming everything's normal in this situation. Now, if we look at what we actually got, first of all, we see males and females affected about equally in every category. Again, that tells us autosomal. But what we don't see, notice that bronze and stunted, there's a lot of those. Red and normal, there's a lot of those. But essentially, there's almost no bronze normal and almost no red stunted. This is not following our 1-to-1-to-1 ratio that we predicted. If you get a cross like that, where the ratios don't match up with the Punnett squares, what that pretty much should always tell you is that the genes for these traits are linked. These genes are found on the same chromosome. The eye color gene and the wing shape gene are linked. And what that means is they tend to be inherited together. They don't independently assort. So what we're saying is that essentially what's going on here is that in that father, that bronze gene that big B and that big S seem to be on the same chromosome. And by the way, it doesn't have to be the dominants on the same chromosome. It could be the dominant and the recessive and vice versa. But because they're on the same chromosome, they're not inherited separately. They're inherited together. So whenever you get a cross and the results don't agree with the Punnett square, realize that probably means there's gene linkage going on. So let's look at the scoring guidelines for cross 3. See what they were looking for. Again, we were trying to answer what conclusions can be drawn from the data from cross 3 and then how the data support that. Number 1, we're going to say the genes are linked. And we're going to say that because the actual cross, our results didn't match our Punnett square. We didn't get a 1-to-1-to-1-to-1 phenotypic ratio. Now, another thing that we might talk about is there's some crossing over going on. And the reason we know that let me get to the right spot here, is if the bronze allele and the stunted wing allele were linked, and the red-eyed allele and the normal wings were linked, we shouldn't get these combinations at all. But we are. And the only way we can get these combinations that we shouldn't get, assuming there's gene linkage going on, is that if during meiosis, sometimes this B gets mixed with that S. It's pretty rare, but sometimes it happens. And sometimes this S gets inherited with that B. So what we're saying is that the bronze eyes and normal wings and the red eyes and the stunted wings, the only way we could get those if gene linkage is going on is that occasionally crossing over creates gametes that otherwise wouldn't be there. And the third option that they give you on the rubric, the genes are 10 map units apart. That's something that most of you probably would never come up with, and you don't need to because you can get all your points from either one of these. But what that's talking about is that essentially how often genes cross over, that's a way to estimate how far apart they are. What they've done here is they figured out that this is about 10% of the offspring. So about 10% of the time the genes cross over, and 1% crossover is said to be 1 map unit. That's a way, again, to estimate distance between linked genes. So these cross over about 10% of the time. So we can estimate that they're about 10 map units apart on the same chromosome. Again, the main thing to get out of cross 3 is if you get results that don't match the 1-to-1-to-1 Punnett square or any kind of Punnett square ratio that you predict, that should probably tell you that the genes for those traits are linked. And then if you get odd organisms that you wouldn't think you should get when linkage is going on, those probably can only happen due to crossing over. All right, that's it for this particular FRQ. Are there questions I can answer for you guys about this one?